This video provides an overview of the autonomic nervous system, presented by Dr. Ali Rizwan.
The nervous system is organized into the central and peripheral nervous systems. The peripheral nervous system is further divided into the autonomic and somatic systems, with the autonomic branch splitting into sympathetic and parasympathetic divisions. The autonomic nervous system consists of motor neurons that innervate smooth and cardiac muscles and glands. It operates under subconscious control to maintain optimal support for bodily functions. Homeostasis is maintained through a dynamic balance between the autonomic branches, often described as a functional tug-of-war between rest and digest and fight-or-flight states. This visual illustrates the distinct pathways and connections of the sympathetic and parasympathetic divisions as they interact with various organs in the body. The autonomic nervous system differs from the somatic nervous system in three key areas: its effectors, its efferent pathways, and how target organs respond. While the somatic system targets skeletal muscles, the autonomic system is primarily responsible for innervating cardiac muscle, smooth muscle, and various glands. Somatic motor pathways consist of heavily myelinated axons. In contrast, the autonomic system utilizes a two-neuron chain, featuring a lightly myelinated pre-ganglionic neuron and a second ganglionic neuron. Somatic neurons release acetylcholine, which is always excitatory. The autonomic system is more complex, as pre-ganglionic fibers release acetylcholine, while post-ganglionic fibers may release either acetylcholine or norepinephrine, depending on the target. This comparison chart highlights the structural and functional differences between the somatic nervous system and the two divisions of the autonomic nervous system. Most visceral organs receive input from both sympathetic and parasympathetic fibers. This dual innervation creates a dynamic antagonism, allowing the body to precisely control organ activity. The parasympathetic division focuses on energy conservation, often associated with D-activities, digestion, defecation, and diuresis. It is most active when the body is in a relaxed state following a meal. The sympathetic division is known as the, quote, fight-or-flight system, associated with exercise, excitement, and emergencies. It redistributes blood flow to muscles and prepares the body for action. Acetylcholine interacts with two primary types of cholinergic receptors, nicotinic and muscarinic. These receptors are named after the specific drugs that can bind to them. Nicotinic receptors are found at motor end plates, on all autonomic ganglionic neurons, and in the adrenal medulla. Binding to these receptors consistently produces a stimulatory effect. Muscarinic receptors are located on all effector cells stimulated by post-ganglionic cholinergic fibers. The binding of acetylcholine here can result in either inhibitory or excitatory effects, depending on the specific receptor type found on the target organ. Adrenergic receptors are categorized into alpha and beta types, each with several subclasses. Generally, alpha receptors are stimulatory, while beta receptors are inhibitory, though a notable exception exists in the heart, where beta receptor binding is stimulatory. Most visceral organs are governed by dual innervation, meaning they receive nerve fibers from both the sympathetic and parasympathetic divisions. Importantly, these two systems do not typically innervate an organ with equal intensity. Dual innervation allows for antagonistic effects. This can occur by having both systems innervate the same effector, as seen in heart rate control, or by each division innervating different cells within the same structure, such as the muscles controlling the pupil. This visual illustrates how the iris of the eye is controlled by both systems, with sympathetic signals dilating the pupil and parasympathetic signals causing constriction. This illustration further details the anatomical pathways involved in autonomic control. This diagram highlights additional functional pathways within the autonomic nervous system. Autonomic reflexes govern most visceral activity, including glands and blood vessels. While influenced by higher brain centers, the hypothalamus serves as the primary integration center for overall autonomic control. Beyond the hypothalamus, autonomic function is influenced by subconscious cerebral inputs from the limbic lobe, as well as signals from the cerebral cortex, reticular formation, and the spinal cord. The hypothalamus coordinates vital functions, including heart activity, blood pressure, body temperature, and water balance. It also regulates emotional stages, biological drives, and reactions to fear or stress. This figure provides a detailed overview of the various levels of autonomic control within the brain and spinal cord. Drugs can influence the autonomic nervous system by mimicking or blocking the primary neurotransmitters, acetylcholine and norepinephrine. Agonists are drugs that mimic and activate receptors, while antagonists are drugs that block them. This classification summarizes how different drugs affect the autonomic system, distinguishing between agents that are cholinergic or muscarinic for the parasympathetic system and adrenergic for the sympathetic system.